Well, thank you, everyone. Sorry once again for those delays. Hopefully we're back online here, and we are delighted to have Edith Pfister from the University of Massachusetts here to present on the latest installment of the HDSA research webinar. And I'm going to turn it over to my colleague, Dr. Laura Fox, who will give some kind of introduction to Edith, as well as instructions for those who may be new to the webinar series as to how to ask questions.
Yes, George, you are going to have to move those slides for me at this point. But yeah, thanks everyone for joining us. Thanks for hanging on. I just want to remind you, for those of you who haven't joined us for a webinar before, or for those who have, there is a way for you to ask questions of the presenter. So if you see in the side there, you can send a question anytime during the presentation. You go to your control panel, and there will be a place where you can type in a question and type it in, and then just hit the send button. And we're going to just be helping to ask Edith those questions at the end. So if you could go to the next slide, please, George. Yep, so, and again, just to remind you, if you want to see this webinar again, or if there's anyone else you know who you think would like to see it, we try to put these webinars up about a week after they first air. And to access it, you can go to www.hdsa.org/researchwebinar, or you can go to HDSA's YouTube channel, which will be at the top of the screen. In that little red circle there, you can click on the YouTube button. It'll get to our YouTube channel, so you'll be able to see this again, and also any past recordings that we have done. So I just want to remind you that there's going to be another webinar next month. We will have a presentation on Wednesday, April 18th from 12 to 1 p.m. Eastern Daylight Time. And that will be about how neural stem cells are being used as therapeutic candidates for HD. That will be presented by Dr. Jack Riedling at UC Irvine. And so without further ado, I would like to introduce our speaker, Dr. Edith Pfister, who received her undergraduate degree from Brown University studying computer science. And after working in that field for a few years, she worked in a lab studying Alzheimer's disease-related genes. And from there, she moved to Neil Aronon's lab at the University of Massachusetts to do her PhD, where she focused on allele-specific targeting of mutant Huntington using SNPs. So that's ways to attack only the harmful form of Huntington and leave the healthy kind intact. And following her PhD, Dr. Pfister returned to Dr. Aronon's lab and continued working on developing AAV gene therapy vectors for silencing Huntington. And now she oversees a lot of the work in Dr. Aronon's lab, as well as managing collaborations with faculty in the UMass Gene Therapy Center and elsewhere. And their labs now focused on Huntington targeting strategies using both gene therapy and synthetic oligonucleotides. So welcome, Edith. Thanks for being here.
All right, thank you very much. Okay. All right, hopefully you can see my screen now. Yes?
Yep.
Okay, so thank you very much for inviting me to do this. This is really exciting for me to get to present some of this research. So I'm hopefully going to make this a little bit fun today, but my basic goal is to give you an understanding of gene silencing technology and the challenges we face developing them as therapeutics in the brain. And I'm going to try to give you a little idea of what the process has looked like for me and for us at UMass. So this is just an outline. What I'm going to do is I'm going to introduce you to our cast of characters. Then I'm going to point out some of the challenges and questions in gene silencing. I'm going to try to tell you how we accomplish this, introduce the technologies that we use at UMass, and I'll give you a little taste of what we've accomplished and where we're going using the sheep as a model system. So our cast of characters. What I'm going to try to do is take you from the big structures down to the small and back up again. What I'm showing you here is a picture, a diagram of the human brain and what are usually considered to be the structures most affected in Huntington's disease. So that's the striatum. Can you see my mouse here? The caudate and putamen. As you can see, these structures are deep within the brain underneath all of this cortex. So now what I'm showing you is a neuron, and these are the cells that are also thought to be affected in the disease. I'm going to talk a little bit about some things where they happen in the neurons. So just to give you an introduction, these neurons have this long process down here called the axon. This is the cell body up here. It's got a bunch of smaller processes called dendrites. And then I'm going to talk a little bit about some things that happen in the nucleus where we have our genetic material and some things that happen out here in the cytoplasm. So I'm going to start with the Huntingtin protein. Hopefully my slide will advance. No. Come on. There we go. So what I'm showing you here is, well, we'll think about Huntington as this nice worker guy. And in people without Huntington's disease, there are fewer than 36 CAG repeats. And this guy is all over the cell. Oops. I'm having trouble getting my slide to advance. There we go. All over the cell doing his job. But when we have a mutation that causes an expansion in the CAG repeat region, he turns into this nasty guy. And he's all over the cell. In fact, he's in so many places, messing up so many things that it's hard to know how to fix it at this point. So the idea behind gene silencing is to intervene before this guy gets out and starts messing everything up, before the protein is ever made. So there are two places where we can intervene to stop the protein from being made, and these are the DNA and the RNA. So the DNA, I told you, it's the genetic material. This is what gets passed down from parent to child. Contains all the information to make the RNA and proteins that the cell uses. It lives in the nucleus, and it's essentially the same in every cell. Now, my favorite is the RNA, which is sort of the workhorse of the cell. And the information from the DNA is copied and processed into RNA called messenger RNA, which I'm representing here. And then this directs the production of the protein. So if you can intervene at this level, you don't get the protein either. So most of the RNA is outside of the nucleus, but some of it is inside. In fact, some mRNAs live predominantly in the nucleus. So the RNA is kind of like the protein. It's all over the cell. So now we have a pretty crowded cell. The DNA and RNA in the nucleus, the mutant huntingtin, stomping all over everything. So what do? So this is my hero. We're looking for this guy. We're looking for something that can come in and fix the problem before it starts by targeting the RNA or DNA. And this is where we get into the idea of gene silencing. So what does this guy need to do? So I'm going to introduce you to the problems and questions in Huntington gene silencing. So I told you there are these two guys in the cell, the nice normal huntingtin and the bad mutant guy. The bad guy, he's kind of dressed up to look like the normal guy, so it can be kind of hard to tell them apart. In an ideal situation, we would want to just get rid of this bad guy and leave the normal guy intact. But it turns out it may be okay if we also, hold on, go away, if we also target the normal. So there's some evidence that neurons will be okay if we have less, at least, of the normal huntingtin. And just to add one additional problem, there are also all these other guys around the cell who look a lot like the normal huntingtin, and we want to make sure we don't target them. So that's the targeting challenge. So that's question number one that we deal with gene silencing. But there's also a second problem, which is the problem of delivery. So what we need is a therapy that gets deep into the brain. So it's as if we're trying to say, this guy, he has to find Wisconsin and Illinois on this map. So there they are. And then if we think about it, it's like there are a lot of bad guys in Wisconsin and Illinois, and we want to get at least half of them. So that's our threshold that we go for, is 50% silencing. And it turns out that we would also kind of like to get, you know, all the other bad guys who happen to be in the rest of the continental US. So that's the second problem, is how do we get these therapies into the brain, and using the US to illustrate that. And finally, we have to think about size. So we do a lot of experiments in the mouse. And what I'm showing you here is the relative size of the mouse brain versus the human brain. Now, obviously, the brain is not a perfect sphere, but you know, the volume, this is just to give you an idea of the volume. So what we need is a bigger brain, because we can get a lot of the mouse brain a lot of the time. So the sheep comes in here. The sheep brain is kind of intermediate between the mouse and the human. And this is why we use sheep. So at UMass, we use two different kinds of sheep. We use normal sheep, and we use this transgenic model of Huntington's disease. So these sheep have the normal sheep Huntington, 2 copies of the normal sheep Huntington, and an extra copy of Huntington. And this is the human Huntington with an expanded CAG repeat. And just for fun, I'm showing you an actual picture of one of our sheep brains. So this brain, well, it looks a little funny here because this is actually a brain that's already been cut. But this brain looks exactly like the normal sheep brain. And for a long time, we thought this was going to be a problem. It looked like these sheep were pretty much normal. And when we're studying a disease, we want to have something that looks somewhat, at least, like the human disease. But we are starting to find that these sheep may not be normal, or they are starting to look abnormal. So that'll be really exciting. When we nail that down a little bit further, we'll be able to use this as a tool for studying the disease, and also for studying what happens when we try to treat the disease. For right now, though, we've been using this model for addressing the gene silencing challenges of targeting and delivery that I told you about. All right, so I've explained the problem, and now I'm going to try to tell you who is this guy? So I'm going to try to introduce you to the technologies that we're using at UMass to do this. So first of all, the basic concept behind all of these technologies is that we're using this property of DNA and RNA, which allows it to be copied. The fact that each base has a complementary base. So blue always matches with orange, and red always matches with green. In this case, though, we're not going to use it to copy. We're going to use it to recognize specific sequences and target them. So I'm going to start with the most recent technology that we've been using, which is a little bit less mature, and then I'm going to end with the more exciting stuff that is closer to maturity. So this is a technology that's been in the news a lot. It's a technology that targets DNA, and it's called CRISPR. So what this is it's a protein in this blob in the background that can cut DNA, and it uses this RNA guide, which is this gray, to recognize complementary DNA and cuts at these little scissors. And so then after the cut, this is the cut DNA, the cellular repair pathways take over and find a way to put the DNA back together. Sometimes it gets put back together just the way it was, but often what happens is there are small deletions or insertions that can inactivate the gene. Alternatively, the DNA can be repaired using another template. But at this point, it's a little bit unpredictable. It doesn't work the same in all cells. So we really are at the point of trying to figure out what happens in neurons and how to best target the huntingtin. So we're on step 1 of targeting. So there are some potential ways we could do this. One of the things that we're thinking about is, can you use 2 of these proteins, put 2 of these proteins on the DNA, and then cut a piece out, and then what happens? Does it join back together the way you want it to, or not? And then alternatively, there are other proteins we could use that perhaps cut only a single strand of the DNA, and then do you get a contraction of the CAG repeat? So these are some of the ideas that we've been exploring. But we don't know yet exactly what's going to happen. And we want to make sure that this is safe, because we're changing the genetic material, we're changing the DNA, and there's really no going back from that. When we do figure out the targeting, there is still the problem of delivery. So in this case, we have to get both a protein and an RNA into cells in the brain. So while this technology is promising, for the most part, what we've been focusing at UMass and what is more mature is an older technology, which is really my favorite. It's not necessarily worse, it's just been around longer, so it's more mature. And this technology targets the mRNA. So again, it's very similar in some ways. This is a combination of a protein, in this case it's an argonaute, and a small RNA that is 21 nucleotides long, that guides the argonaute to the mRNA and then cuts the mRNA. So you don't correct the gene at the DNA level, but you eliminate the mRNA before you get a protein. So when I first started at UMass, we were at the place in this technology, I think, that we are now with the CRISPR, where we were trying to figure out the best way to target Huntington. So imagine you have this normal Huntington, the good guy, and the bad guy in the cell. But you want to distinguish between those two. But the challenge is you can't use any of the obvious characteristics. So you can't use the fact that he's green or has ugly teeth or has 4 arms. You can't use those. What do you do? So it turns out if you have another change that's not one of the obvious characteristics, something like a green glove versus a yellow glove, and you can figure out which guy has the yellow glove, you can target him. So that's essentially what we were doing. We were using these single nucleotide differences that were far away from the CAG repeat expansion. We were figuring out which one was associated with the expansion, and we were targeting that. In the meantime, it turns out that we also thought that perhaps we didn't need to be so specific. So we also were developing a RNA guide that could target both the mutant and the normal Huntington. So that's where we were at that point. But we still had a delivery problem. And at the start of this, we really weren't doing delivery very well. We were using a basic unmodified RNA, which can work very well in cell culture, and I worked a lot in cell culture. But it turns out that this RNA doesn't get into the cell by itself very well. So if you put it in the brain, a lot of it ends up sitting around outside of the cell, not just completely useless. Additionally, this RNA is unstable. So there's a lot of proteins, the orange blobs, that chew this RNA up. So basically, when we tried to do this in the brain, we weren't actually getting very much activity. So at that point, we switched to this gene therapy type approach. So what we're using in the gene therapy approach is a modified AAV virus. So AAV viruses, they're very common. The natural AAV, they're non-pathogenic, so they don't cause any disease. The natural AAV is a small virus that contains genes mostly to help it make more of itself. What we do in gene therapy is we take out those genes and we replace them with what we want to put in the cell. And this gets into neurons very well. So in a neuron, what happens is it gets processed, and we can make something that looks a lot like RNAs that are already in the cell, and then this will be loaded into the cellular argonaut, and you can cut mRNA. And this is actually what we've recently put into sheep. So what I'm showing you here is an injection into one of these sheep. So what we do when we do these injections is we put a contrast agent in so you can see the spread of the material. So the spread is around this red dot. There's sort of a white area, so that shows you the spread of the liquid, basically. And as you can imagine in the sheep, this is the brain. So you can, in the sheep, this is actually covering a lot of the striatum. So we inject here into this slice that's we're calling 2M1. And what we see is that when we inject the AAV there, we get 50% reduction in human huntingtin in the caudate and putamen in that block. And this silencing spreads to the block in front of that. So this is the front of the brain, and this is the back of the brain. This is the anterior block, 2A. So silencing spreads to 2A, but it doesn't spread to this one in the back, the posterior. So we've made progress here, but as you can imagine, the striatum in people is much bigger. So there's still room for improvement. And so now we come full circle back to my favorite, the non-viral delivery. So we had a colleague join us who knows a lot about more about chemistry than I do, and her lab has been working to improve the stability and delivery of oligonucleotides. So this is a modified siRNA. So what it is it's designed to look like an RNA, but it really doesn't have any more RNA in it. But the cell recognizes it as RNA and processes it as if it was RNA, so it can load into argonaut. So what we do when we look at these look at the distribution of these is we put a fluorescent dye onto this molecule, and that's represented by this red star. The fluorescent dye is red. And we test it first in mouse. So this is a normal mouse brain. It's very white, doesn't look like much. And then we inject this fluorescently labeled oligonucleotide into the mouse brain. And what we see is this beautiful pink brain. So you can see here, right, you can actually even see the injection site here. Now, I have never seen delivery like this before. And I realize this is still a mouse brain, but I have never seen anything that really goes outside of the striatum like this. So this is amazing. The next step of doing this is to do the same thing we did with the AAV with this chemically modified oligo. And that's where we are today. So what we're doing right now is we're putting this modified siRNA into the sheep brain, and we're starting with normal sheep, and we're looking at the distribution in the brain. So I was not able to pull a picture for this one, but what I can tell you so far is that it looks very nice. The brain there is fluorescent in the brain, and the only question now is, are we getting these deep structures in the striatum? And so that actually is the end of my presentation. I hope that I have accomplished my goal of giving you a basic understanding of gene silencing. And I just want to thank all of these people who worked on this. This has been a huge project. We had a lot of authors on our sheep paper. The Aronin lab, where I work, the DeFiglia lab, who does a lot of the protein work, the Mueller lab did a lot of the viral stuff, and Anastasia Kavarova, whose name I can't pronounce, her lab does the chemically modified siRNAs, and Miguel Sena Estevez is one of my mentors. And then we also had help from Australia and New Zealand. So thank you very much for listening. And I guess I can answer questions.
Yeah, thanks. Thanks, Edith. Can you hear me okay?
I can.
Great. Thank you so much. So yes, folks, if you're hopefully still on the line and you do have any questions, feel free to type them in the question box at any point. I have a few questions. Touched on your last slide, you know, kind of the issue in Huntington lowering gene silencing, whether it's with CRISPR or AAV or oligonucleotides, is delivery and spread. And these are things you showed nicely that in some ways what you're doing to kind of get at that tough hurdle or attack that tough hurdle. Are there plans in the sheep to look at some of these newly modified viruses that have been created by the field in particular? I don't know if you're familiar with the PHPB virus that was created by Ben Deverman at Cal Berkeley, I believe, that they've shown some pretty interesting spread upon intravenous administration of the viruses, so kind of avoiding having to do an intrastriatal or brain injection of the virus. You thought,
yes.
Thought about
yeah,
doing that
so we had a plan to do it in sheep, and what happened is it turns out that these may be highly mouse specific. At least the PHPB and PHPEB seem to work in some species and not in others, like particularly in mice. So that was kind of the hurdle for that. I think Miguel Estevez is actually working on this, and so I think what he's seen is that you get an increase in transduction in mice. It doesn't work quite as well in, I think, cats and rabbits. So the question is whether it's just species specific and whether there's a way to use the same methods to develop something that would work in a non-human primate, for example, because maybe you don't really want it in sheep in that case. If you're going to find something species specific, you want it to be closer to human.
Great, thank you.
Maybe I could jump in with a question as well. This is Leora. I assume everyone can hear me. I'm wondering if you could touch perhaps on how this technique might differ from the kinds of strategies that are currently in the clinic, and what it would mean for delivery in humans.
So are we talking about antisense or?
Yeah, antisense.
Yeah, so I mean, so the modified oligos are actually very, in some ways they're very similar to the antisense, in that you're trying to make something synthetic that works in the brain. I think, delivery has been the problem for that as well. And so I think that's where Anastasia has really focused her effort, is getting much, much broader delivery. So I think that that's going to be the main advantage. I think one of the things that I can potentially see happening is if we see delivery to the cortex with an oligo, but we don't get deep into the striatum, like maybe you combine something like an AAV for the direct that you only have to do once into the striatum, and some sort of oligo that you can put into the CSF, either antisense or siRNA. The siRNA is, I just think in some ways it's an improvement on the same strategy. They're different in that the antisense is working maybe more in the nucleus, and the siRNAs are working in the nucleus and the cytoplasm, but I think it's going to be an advantage with delivery at this point.
Thanks.
A few questions coming in here from the audience, Edith. One is, have you noticed any damage to the brain at the injection sites?
So I've in our initial AAV work in mice, there were some of our AAVs caused extensive damage in a mouse brain. When we put that into a sheep brain, we didn't notice anything at all. So I don't, there's an interplay between the volume that you're injecting and how it spreads and toxicity. So we haven't noticed much with the oligos, and in the sheep brain, I really have never seen any damage at all.
Great. Another question from the audience was that given the extensive distribution you're seeing, at least in preclinical models, in the mouse, and hopefully, although we didn't see it, but you said it looking really promising in the sheep, do you think you'd ever be able to deliver these modified siRNAs or viruses into the CSF like what's being done with the oligonucleotides, or will they just never penetrate the cells?
Yeah, so actually, I didn't say it, but we are delivering the oligos in the mouse into the ventricle, but the idea is in the sheep to put it in the CSF. So you don't have to do direct intrastriatal delivery of the oligos.
Okay, so for your modified siRNA oligos, that work that you are that's now underway to turn the white brain pink or red,
yeah,
that was intrathecal or CSF administration.
Yeah.
Okay. Another question, and I don't know if you could comment on it, but I'm pretty sure there's some members at UMass Worcester that are working on this topic, is related to viral expression of these silencing reagents, these siRNAs. One of the, I guess, the perks of or benefit or pro of this approach is that it's in theory a one-time administration, meaning that you dose these animals and hopefully humans one time, and the viruses will express the silencing reagent forever. And I could also see that as a huge con, a huge something, red flag, that's something we have to take a look at, because once this drug or virus is administered, it's going to express forever, and there's no shutting it off. What work's being done to to develop regulatable AAV viruses that can either be turned off or turned on with maybe the administration of an antibiotic or something like that?
Yeah, so there are people working in the Gene Therapy Center on regulated promoters. So, and that work is being done, something that can turn it on. The sort of the drawback of that is usually you're having to express more things to actually regulate it. So you're still expressing something indefinitely. It's just not the siRNA. But there is work being done on that, so we should have, and it may allow you to tune the expression of your siRNA in that case. So if you get too much expression, if you can just treat with less antibiotic, or the one that's popular, rapamycin is very popular, that could have a benefit. Definitely an avenue that's being explored.
Great, thank you. I think another question, I'm going to paraphrase it, is, maybe you could speculate on, you mentioned about the viral delivery, and then you touched on the the modified siRNA oligo delivery. What do you think, delivery into humans, what do you think is the most attractive approach right now, based on your understanding and work? Is it delivery using AAV, packaging the silencing reagent, or is it delivery directly into the human brain without a virus such as the modified RNA?
Yeah, so I actually go back and forth on this, but my feeling is that the AAV is more mature. There's other diseases that have made a lot of progress in that regard. So in some ways, I think that might be the first strategy. But I think that the modified siRNAs may, in fact, sort of leapfrog that because they are looking so spectacular. And because you put it in and it's not permanent, like they may be, it turns out they may be fairly long-lived. They may last, 2-3 months, but it's not a permanent modification. So in some ways that's really attractive.
Yeah.
Especially if you can deliver them into the CSF rather than directly into the brain. I mean, there's so many reasons to think that that technology may leap over the viral delivery.
And do you have data on the lowering of Huntington? So the approach, and maybe you mentioned this, and I apologize if you did. So the approach being currently underway in the Huntington's disease sheep, is this are you looking at an allele selective approach, or are you?
No, those are non-allele selective. Yeah,
Okay.
so my experience with the allele selective in the viral is that it's not so selective. So it works better in an siRNA context. So right now in the sheep, it's not allele selective, and we only have one allele in the sheep to look at anyway. Although it doesn't target the sheep huntingtin. So in the sheep, it sort of is allele specific in that, they still have their normal sheep huntingtin. So I guess in that case, we don't know what would happen if you lower everything. I think we might learn that from the oligos more, because actually I didn't mention it. It was in my notes, but I didn't mention it, that in the mouse brain, we see like 80 to 90% silencing, which is more than we see with the AAV.
Fantastic. I don't think there's any more questions that I see coming from the audience. I don't know, Leora, if you have anything else to add?
No, just want to thank you again very much, Edith, for sharing that with everybody and for making it clear and with fun cartoons. So thanks again.
Thank you.
Thank you, Edith. I appreciate you taking the time, and hopefully you stay safe up there in Boston with the snow. And thank you all for bearing with us with our audio issues. We're going to look into that, so hopefully it doesn't happen again. But please do join us next month, as Leora said, with Jack Riedling from UC Irvine talking about stem cells, another exciting topic. And until then, wish you all a great week, and hope to talk to you guys soon. Take care.